This video lecture is about choosing a statistical tool to test the hypothesis made by the researcher. In terms of selecting a statistical test, the most important question is, what is the main study hypothesis? In some cases, there is no hypothesis. The investigator just wants to see if what is there. For example, in a prevalence study, there is no hypothesis to test, and the size of the study is determined by how accurately the investigator wants to determine the prevalence. Therefore, if there is no hypothesis, then there is no statistical test needed. This is a table to give us a glance on what are the different statistical tests needed. So our statistical tests or tools is dependent on the type of data variable that we are going to gather. So the data variable, such as nominal, is appropriate to be processed using McNemar's test. Ordinal, which can be ordered categories, can be processed by Wilcoxon, and quantitative variables also can be processed using Wilcoxon. And quantitative normal distribution datasets can be tested using paired t-test. These are just some of the statistical tests that can be done. Remember that the test depends on the type of variable that you can gather from your research instrument. This next table is another quick glance on the different tests according to input variable and outcome variable. In our input variable, we have nominal, categorical, ordinal, quantitative, discrete, quantitative non-normal, and quantitative normal. When you say non-normal and normal, it means the distribution of the data sets. In the outcome variable, we have also the same nominal, categorical, ordinal, quantitative, and both non-normal and normal. So once you have checked what is the type of your input variable and outcome variable, you can determine the most appropriate test that can be done to your study. So if, for example, your input variable is a nominal and with your outcome variable is also nominal, then you can use t-test. Well, if your input variable is categorical and your outcome variable is quantitative normal, then you can use analysis of variance. This part here is determined by your statisticians, but what is important is, as you are a pharmacist, you have to understand how and why your statistician chose that kind of statistical tool. Another consideration in choosing statistical tests is if our data is parametric and if your data is non-parametric. So there are statistical tests that are appropriate for parametric data sets and appropriate statistical tools for non-parametric. So your paired t-test is applicable for parametric data sets, and if it is in non-parametric, you will use Wilcoxon rank sum test. Also, if we have non-parametric Mann-Whitney U-test, we will have unpaired t-test for parametric side. To compare a relationship for parametric tests, we will use Pearson correlation, while for non-parametric equivalent, Spearman correlation is used. If we have to compare the difference between three groups or more, then we will use one-way analysis of variance for parametric datasets, and non-parametric datasets we will use the Kruskal-Wallis tests. Now let us have the first statistical tool to test for hypothesis. If our hypothesis needs to be tested between two independent groups, we will use sample t-tests. This is useful for parametric datasets to check if the hypothesis is true between two independent groups. This test uses mean and standard deviations. So what are we testing for t-tests for independent samples? Given that your null hypothesis states that the two groups means are equal, your research hypothesis will be stated as there's no significant difference between the two groups. For alternative hypotheses, you assume that the two groups are not equal. Therefore, your research hypothesis that can be made will be there is a significant difference in the two groups. Now, for the decision rule, we will reject the null hypothesis if the p-value associated with the computed t-value is less than 0.05. We do not reject or accept, in other words, the null hypothesis if the p-value associated with the computed t-value is greater than 0.05. Let us have an example. In this research, a study was conducted by a group of fourth-year pharmacy students in XX school to determine the level of male and female community pharmacists' awareness on antimicrobial dispensing. A standardized survey questionnaire was adopted from an international research in a 5-point Likert scale. With that research topic, we can create a relationship of the variables in this manner. Based from the study, we will have two types of variable, the independent variable and the dependent variable. So the independent variable is sex, which is in two categories, your male and female. And then the sex will determine, or it is the, the determinator of our level of antimicrobial dispensing. The knowledge might be different from male versus female. Now, in that case, the problem can be stated, or the research question can be stated this way. What is the level of awareness on antimicrobial dispensing of female community pharmacists? The second is, what is the level of awareness on antimicrobial dispensing of male community pharmacists? For the third statement of the problem or research question, this is the time you will state your hypothesis in a manner of research question or statement of the problem. So it will be stated this way. Is there a significant difference in the level of awareness on antimicrobial dispensing between male and female community pharmacists? So for number 1 and number 2, SOP, you will use descriptive statistics, mean and standard deviation. For the third research objective, since this is to compare significant difference or to test their hypotheses, it will involve inferential statistics, which is t-test for independent samples. So the independent sample t-test should have the following assumptions: the normality of the distribution, the equality of variance, there's independence of observations, there are no outliers, and it is done using random sampling. And there is IV, which is independent variable and dependent variable group. So here in this slide, this shows us the result that can be generated from our statistical software such as SPSS and JASP. So for independent sample t-test, you will be given with a table that looks like this. So for independent sample t-test awareness, the value or the p-value there here should be less than or equal to 0.05 to test for significant difference. Now, the value given in our results is 0.974. That means to say it's more than our desired p-value. Therefore, our judgment will be there is no significant difference in the level of awareness of our female and male pharmacists. In the next table, we have here assumption checks. For assumption checks, this is used to test for normality. So for the p-value of higher than or more than greater than 0.05, that means the distribution of the dataset is normal. So with the value given or generated by the software, it has a p-value of 0.3 and 0.39. That means it is more than or higher than 0.05. It means to say that the data distribution is normal. Now for test of equality of variance, the p-value should also be higher than our 0.05 in order for our dataset to be considered homogeneous. So with this set, our p-value for test of equality gives us 0.498, which gives us a dataset that is considered as homogeneous. This time, we were going to present our data. This table shows that male community pharmacists have relatively quite higher level of awareness on antimicrobial dispensing as compared to female with a difference of 0.005. The t-test for independent samples confirmed that the difference in the level of antimicrobial dispensing between male and female community pharmacists is not significant. That is how you will present the data that was generated by statistical softwares.